Now, obviously, that doesn't cover all the stuff we might want to transport across a membrane. Like, what about even bigger polar stuff? Or, heck, we keep moving things from high concentration to low concentration. What if we instead want to move against that concentration gradient and go from low to high? Enter active transport. So the reason we call it active is because it requires energy in the form of adenosine triphosphate, or ATP, which we've mentioned a couple times now. Now, there are kind of two broad approaches to active transport. So things that are moved by membrane proteins come through the phospholipid bilayer, similar but different to the membrane proteins involved in facilitated diffusion. Or you could also do something called bulk transport, which actually involves vesicles, and we've kind of talked about it already. But let's start off with kind of membrane protein example. Specifically, protein pumps or carriers, okay? So this is kind of similar to the facilitated diffusion that we just talked about a few moments ago, in that there are proteins kind of transcending across the membrane, transmembrane proteins, and they move things across by changing their shape, right? Really similar to facilitated diffusion. However, unlike facilitated diffusion, you need help to do this, right? They need help changing their shape. In this case, help in the form of ATP, or adenosine triphosphate. Also, unlike facilitated diffusion, they move things against the concentration gradient, going from low concentration to high concentration. So, how do they actually utilize this ATP to do the shape change? Well, they do something called coupled reactions, where you pair a reaction that requires energy with a reaction that easily gives up energy. In this case, it's really easy to utilize the energy stored in the bond between the kind of second and third phosphates of adenosine triphosphate by kind of snapping off that last phosphate and attaching it to, say, the membrane protein. This means you've phosphorylated the membrane protein, which then changes its shape and does its thing. Now, a great example is the sodium-potassium bump. And by bump, I mean pump, which basically ships 3 sodium ions out of a cell and 2 potassium ions into a cell, and requires ATP to do it. Simply put, when the phosphate from the ATP binds to the membrane protein, 3 sodium ions from inside the cell also bind. And then the shape of the protein changes and the sodium ions are kicked out. When they're gone, 2 potassium ions bind to the pump, and when that phosphate from the ATP that was attached on the inside, when that gets released, the protein changes shape again, and those potassiums get ejected into the cell. So, there's a lot. To summarize, this is the sodium-potassium pump. Kicks 3 sodium ions out, brings 2 potassium ions in, but requires energy in the form of ATP to do it. Okay, I may have gotten a bit carried away with that description, but that's because you're actually going to see these sodium-potassium pumps back in 116 or later in 116. They're going to come and haunt you, and we'll be talking about them a lot and how they are involved with the neurons and how neurons function. But so just keep those in mind for the future. Finally, another category of active transport is bulk transport, where you're, unsurprisingly, moving large numbers of often large molecules at once. Unlike the other forms of active transport, membrane proteins aren't necessarily involved, which means the things being brought into the cell do not actually cross the plasma membrane. Instead, they get brought into or out of the cell using those vesicles that we keep talking about, right? So they're still considered a form of active transport because it definitely requires energy in the form of ATP. Now, there are actually several different types of bulk transport, which is what we'll be talking about for the rest of the lecture. The first type of bulk transport is exocytosis, where things are being released from the cell. Typically, these things include waste or the proteins, maybe made by the ribosomes, folded by the RUFER, and packaged by the Golgi, and then secreted through the membrane. There's also some other kind of secretory products that can be created and shipped out by these vesicles. Now, the vesicles containing these materials, like the folded, modified proteins, fuse with the plasma membrane and then kind of release its content as it fuses. Conveniently, fusing these vesicles with the plasma membrane also increases the size of the plasma membrane by adding more phospholipids. So if you're trying to grow your plasma membrane, the primary way to do it is by adding some vesicles to it. Cool. Of course, you also may want to bring stuff into the cell as well. Enter endocytosis. Rather than merge a vesicle with the plasma membrane to kind of kick stuff out, you create a vesicle to kind of butt off and bring stuff in. Now, there's actually 3 different types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Okay. Now, phagocytosis is considered cellular eating, and it's when a cell kind of engulfs a large particle, like a really big protein maybe, or maybe even a smaller cell. Now, some cells actually extend a portion of their plasma membrane out and around kind of these large particles, and then pinch off the vesicle to then get released inside of the cell. Similarly, pinocytosis is called or considered cellular drinking, and it's when a cell intakes fluid and the dissolved materials in that fluid. In other words, lots of little stuff suspended in a fluid, right? It does this by making a vesicle and then kind of pulling the fluid in, kind of like like pulling the sides of a plastic bag, causing like a vacuum or something, so then it sucks all that fluid inside. In both of these cases, though, this cell is kind of just grabbing whatever it can, right? In other words, both phagocytosis and pinocytosis is very random, non-specific, right? Receptor-mediated endocytosis is almost literally the opposite of random. Instead, there are receptors or receptor proteins in the plasma membrane that bind to specific macromolecules. So you have a specific protein for a specific macromolecule. When these macromolecules attach to their associated receptor protein on the outside of the cell, the plasma membrane will then eventually fold inwards and bring the protein receptors with the attached macromolecules into the cell in a vesicle. To kind of help this process, there's actually a layer of proteins that will bind to the inside of a plasma membrane to kind of provide some structure and rigidity to the plasma membrane as it does the folding, and then the vesicle itself. A quick note here, the book, and by the book I mean OpenStax, names these structural proteins as clathrin. Don't sweat it so much. We don't really talk about it too much. The moral of the story is, though, that these vesicles that get formed from particularly receptor-mediated endocytosis are what we call coated vesicles, okay? As a side, or directly related to this, really not an aside, the receptor-mediated endocytosis happens to be kind of the major way that you bring macromolecules into a cell. which kind of makes sense, right? You really only want to bring the stuff that you specifically want into the cell, right? You don't want to just kind of grab anything willy-nilly. So, yeah, there we go. Oh, quick note, sorry here. That's it should not say figure 7.19. It should say like figure something else. What figure should it say? It should say... I'll leave it in the comments, and I'll leave links, because I have to figure it out. That's an old figure number from the wrong book that we don't use anymore. So, whoops. I'll include the figures in the comments of the video. Okay, anyway. Woof. Next lecture, lecture 7.1, we'll be talking about metabolism and energy change. So, we're going to be talking about the basics of metabolism. We're going to be talking about what happens with free energy change. And then we'll finally talk about ATP, right? The adenosine triphosphate that we've mentioned about like a million times. Next lecture. Get excited for it. I know you are. Before that, though, here's lecture question 6.2 to leave you with. Which of the following are true regarding cell transport? Good luck. I'll see you next lecture. Lecture 7.1. Bye.